So today I'm going to tell you really a bit about an interesting side project in the lab that mainly was done by Ivan Gando and Manuel. And this is how we came up, actually through a collaboration with the Tim Cardozo laboratory, how we came up with a compound that opens KTP channels. And I just wanted to tell you the story. And so Colin has made my life really easy today because he already introduced the ATP-sensitive potassium channel, and I don't have to do that. And he mentioned to you that unlike other potassium channels where you've got 6 or 4 transmembrane domains, these are inverse rectifier potassium channels with only 2 transmembrane domains. And these are the different subfamilies, a very diverse group of proteins. So the KTP channels basically form into this category over here. And of the inward rectifier potassium channels, there's something like 15 or something, I've lost count. And they fall really into 4 different groups. The KIR3s code for the G protein activated potassium channels. Those channels that get activated by acetylcholine and slows the heart rate when you sleep. The ATP sensitive potassium channels we're going to talk about today, you've heard from Colin really, there are 2 of them, KR6.1 and KR6.2. And Colin has been mainly talking about the KR6.1 channels in Cantu syndrome. The KR1s are really involved in potassium transport, mainly in the kidney, and then the KR2s are responsible for the inward rectifier potassium channels, such as in the heart cell or neurons, that causes a resting potential, or negative resting potential. I'm going to talk briefly about all 4 of these subfamily members. As Colin has mentioned, the KTP channel is formed by not only the KR6, 4 of the subunits, but also 4 of the SUR subunits. And Colin has also explained to you how they're regulated by ATP and ADP, and I'm not going to go very much into that. So the KTP channel has diverse functions. There are actually many classes of ATP-sensitive potassium channels in our bodies. And one of the best known is really the role of the KTP channel in insulin secretion that Colin has alluded to as well. And so in the pancreatic beta, in islands in the beta cells, under the basal state, these channels are open, and they basically set the resting potential to be fairly negative. When they get closed after lunch, like we just had, then the membrane potential depolarizes. The beta cell starts firing, and during this process, what's going to happen is that the insulin vesicles is going to release the insulin. And that's just an example of, one example of KTP channels function in our bodies. But as I said, these channels are really diverse, and they exist in blood vessels, endothelium, smooth muscle, and you've heard how gain-of-function mutations and loss-of-function mutations of the KR6.1 can actually cause major changes in blood flow. You've heard just now that insulin secretion in the pancreas is regulated by these channels as well. In neurons, they play a really important role in neuronal excitability, and there's even evidence that they might help release some of the transmitters in some of the sympathetic and parasympathetic neurons. A lot of interest in the heart, how opening of the KTP channels in the heart can lead to cardioprotection. Then there's also a subclass of KTP channels, which is probably a different kind of channel in mitochondria that's really gotten a lot of interest in the literature as well. The purpose of this slide is to show you that there are indeed, as Colin has pointed out, a number of openers and blockers of this channel, but they are not very specific to any one of these subclasses. You can see, for example, Rogaine was developed as an antihypertensive agent, but it actually caused increased hair growth and is now marketed as a hair growth agent. And there are drugs, as Colin has pointed out, that are used for helping to regulate insulin secretion. But in general, one would ideally want to have a drug that is specific to a specific subclass of KTP channels. You've heard how the channel is regulated by ATP and ADP. It's blocked by intracellular ATP, and that the drugs normally bind to the sulfonylurea receptor. What you've not heard so far is that the channel is also regulated by PIP2, and this is actually something that was discovered quite a while ago by Colin and others. And that PIP2 can actually activate the channel by binding to particular residues in the KR6.2 subunit, and then kind of decouples it from SUR and activate the channel, right? So the story I'm telling you today started off with this guy. So Huaqian Yang was a postdoc in my lab. He's now got his own lab in China. And he basically found that KR6.2 can be regulated by palmitoylation of a particular cysteine residue, cysteine 166, I think it was, in the KR6.2 subunit. And I frankly didn't understand how it is possible that that palmitoylation of that particular residue can lead to opening. My natural thinking was it's going to increase the G-loop gate, and therefore it's going to have a higher open probability. So we then basically went to the lab of Tim Cardozo, who I believe is here in the audience today as well. He's a computational biologist. He's doing a lot of structure activity kind of studies. And we asked him, and Will Nally in particular, to really find out, what causes this increase in open probability. And through their work, that was again validated by Huaqian, found that palmitoylation actually regulates the PIP2 sensitivity of the channel. With a palmitate in a particular place, the affinity for PIP2 seems to have been increased, and therefore you can get better opening of the channel. That was what these data suggested. So following on from that study, Tim really focused on this particular pocket. This is where the C166 is the palmitoylated and PIP2 in this area. Tim really started looking a little bit more at this pocket, and he wanted to know whether this could be a potential druggable pocket. This is what he does, right? And so at the time, there really wasn't a structure for KTP channels, and certainly not in the open state. So Tim has basically made models of the KTP channel, both in the open state and the closed state, and used those models to get a better atomic resolution kind of model. And then what he did was to perform a virtual library screen. And they screened 500,000 compounds against the Cambridge library to find whether it put any compound that could potentially bind in that pocket. They've identified several of these compounds and ordered the top 15 of these heats. So you've heard about this ion lab, and I can just tell you that this is something that I'm involved in as well. But the main guy that's really involved is Suman Song. He's the manager. And the kind of instruments we have here, shown here, ranging from brain slice recording systems, regular patch clamp systems, medium throughput planar patch clamping, and measurements of transport activity. But we also have this guy in there, which is really just a plate reader with liquid handling abilities. And we've used that system over there to use a thallium flux assay. And Ivan then has developed this thallium flux assay to screen whether these compounds can actually affect the KTP channel. So the way it works, basically, you've got a fluorophore that goes into the cell. Thallium is not a normal cation. It doesn't exist really in our bodies. But when you add it outside, thallium goes through potassium channels better than potassium does. And so when thallium goes in, you get fluorescence. And we basically measure the fluorescence over time with this particular device over here. What you see here is a stable cell line that's been put in this flex station. And we're looking at panacetyl as a positive control. Panacetyl is a known, well-characterized KTP channel opener. And when panacetyl is added in red over here, upon the addition of thallium, you can see a very, very steep increase in intracellular thallium fluorescence, showing that we've got a valid method. And then several of the compounds have been used. Many of them actually had an effect on the KTP channel, but we were particularly interested in this one over here that seems to activate the channel almost as well as pinacetyl. Now, this initial screen was with 100 micromolar of each compound because we just wanted to see whether there was an effect. On this side over here, you're looking at the thallium flux rate and the different compounds, and you can see that several of them had relatively minor effects versus DMSO, pinacetyl in red, and this particular compound as good as pinacetyl. We verified this with patch clamping. So this is a whole cell patch clamping, again, of the same cells, stable cells, expressing KRAS 6.2 and SUR2A in this particular case. Apply the compound, and you see a huge increase in current with this compound. That can be blocked by the sulfonylurea clobentlamide, as you've heard previously today. And this compares the amount of increase by pinacetal in this compound, and it's very similar. What Ivan then has done was he ordered structural analogs of this particular compound that I'm shortening here to 2985. If I had to write the name of this compound, it'll go from this slide to this slide. So I just made it 2985. And you can see that several of these, actually you can see it probably better in the bar graph over here, activated the KATP channel, but not nearly as well as the 2985 that you can see over here. The next obvious thing to do is to look at concentration responses, right? And so he did that. And basically here we again look at thallium fluxes with pinacetal, different concentrations, as you can see here. And with this compound, different concentrations. And if you plot the dose versus the rate, then you can see here that pinacetal, so EC50 probably in the region of about a micromolar, whereas this compound is more like 5 micromolar, but still in the low micromolar range, which is kind of interesting. Of course, now what we needed to do is to test it against other channels, right? And this was interesting because we tested each of these groups of channels. The KR2 channel is here expressed in HEK293 cell. We did whole cell recordings. And what we're looking at here is just a HEK293 cell that doesn't have the channel, transfected with KRAS 2.1. We saw a robust current. And this compound at 100 micromolar had zero effect on KR 2.1 channels. This study here was done by a colleague of mine in Japan, Tomoe and Ishitani. They've got a Xenopus oocyte system over there where they inject a combination of KR 3.1 and KR 3.4 into oocytes and then basically look at whether these G protein activated channels are affected by a compound. Here we used a concentration of 15 micromolar, which is 3 times the EC50. And you can see here it literally had no effect on the current at all. And you can see, well, I'm not going to go through this. I think you get the message. Now, this to me was the most interesting and surprising result because KR 6.1 sequence-wise is very similar to KR 6.2. A lot of similarities. When we look at the effect of KR 6. Of the drug on stable cell line, expressing KR 6.1 and SUR2B, which is the vascular type of channel, we can see that pinacetal can activate this channel, and this compound really didn't. Your DMSO is over here, and none of the compounds really activated the KR 6.1 channel. Some of them actually even blocked the KR 6.1 channel, which we have ideas of why it might do so, but I'm not going to go into that right now. So how can we use this compound? So this was an idea that was given us by Xiaolin. And then the question would be, can this compound correct patient mutations where in KR 6.2 that causes a decreased open probability? And we tested 3 of those. F155L, T62M, and R301C. And in the next slide, you're going to see some thallium flux data again. And you can see that the wild type, in this case, is expressed together with SUR1 because we wanted to recapitulate the pancreatic channel in this case. It was activated by, or flux occurred during the assay, and diazoxide activated the channel, right? Of these compounds, of the mutations, all of them had a reduced thallium flux, as you would expect, if it reduces the open probability of the channel. But only one of them was activated by this compound, 2985. Now, while therapeutically this is not good news at all, because there's only really one kind of patient you can potentially treat with this, it might tell us something mechanistically, which is interesting, because we know that the residue 301 is actually involved in PIP2 sensitivity as well. This is a study from, I believe this is Xiaolin's study, showing that if you look at the surface expression of KATP channels and the effect of the mutations on at the position of 301, we can see that 301C that we looked at really doesn't have too much effect on the surface expression relative to some of the other mutations. However, when you look at the current, this is the current that can be blocked by ATP in a reversible manner in wild type. In the 301C, when the patch is pulled off, the current activates, but it's immediately run down again. Now, this mechanism of rundown has actually been studied before. It's thought that this rapid rundown is actually involving PIP2 binding. And the reason for that really is because you can reactivate the channel with long exposure of high concentrations of magnesium ATP as thought to stimulate the PIP2 generation. So this gave us food for thought and ask whether this compound actually can act by substituting for PIP2. The assay that we've used is actually one developed by Noma's lab many, many years ago, where they basically study the channel with excise patches and then apply calcium. And the idea here is that calcium activates phospholipases, phospholipase C, that breaks down PIP2, right? And you can see this here. This is not our data, this is Noma's data. See the channel activity over here that's blocked if one adds the calcium, and then magnesium ATP can restore this effect. In the presence of Wortmannin, this doesn't happen. Wortmannin basically would catalyze the reverse reaction and actually replenish the PIP2, right? If they do it again with, or kill the channel again with calcium, potassium ATP doesn't substitute for it, but magnesium ATP does, thinking that this is actually the mechanism that's being studied here. We've actually done some of this ourselves in the lab too, and not only us, others have also done this. When you actually apply PIP2 after the calcium, you can see the calcium over here, channel activity goes away. When applies the PIP2 directly to the channel, it also activates the channel, suggesting that this could be a potential readout for measuring the PIP2 sensitivity of the channel. And this is the experiment that Ivan has most recently done. Recording of CAR 6.2 SUR2A stable cells. And here you can basically see channel activity. Apply the calcium and the channel activity goes away, right? You can reactivate the channel with incubation of magnesium ATP, just like Akinoma has shown, right? He does it again, apply the calcium, and now apply the compound. And you can see that the compound very powerfully activates this channel, suggesting to us that perhaps that there could be a PIP2-dependent mechanism. Now, what you're looking at here is an amateur attempt at doing some molecular docking. Tim, if you're here, you're going to have to repeat this, please, because I'm not sure what I've done here. But at least when I'm doing some in silico docking, we can see that the PIP2, this is blind to docking of the CAR 6.2 in the open state. Blind docking, the program automatically finds pockets and then docks compounds in there, docks PIP2 in the appropriate place, where the binding is coordinated by residues that we know are involved in PIP2 binding. We repeat the same thing with this particular compound. 2985 binds to exactly the same pocket with similar residues that coordinate binding. This makes us think that maybe this compound 2985 could be a CAR 6.2 specific PIP2 analog, possibly. So we are now in the process, because we recently have this technology in the lab where we can do higher throughput electrophysiology recordings using this instrument over here. And this instrument has really added another level for the services that Iron Lab can offer. And this technology, I think, is quite good and interesting. So using that, the latest experiment Ivan has done was to look at sodium currents in these cells, TE671, which I believe expresses a KV, or NAV 1.3 maybe, or something of one of the NAVs. And found that, these data were produced in one afternoon. So you can see that in 25 cells over here, that before and after the compound, there might be a slight inhibition. This is 100 micromolar. We're now repeating this with more relevant doses, and we're also going to start looking at other channels to see if this compound affects the other channels as well. So in conclusion, then we've identified, or Tim has identified a novel druggable pocket in KR6.2. He's identified at least one compound that is a channel opener. And to my knowledge, this is the first and one of the only KR6 or KIR subfamily specific opener identified to date. So ongoing is with ongoing screening to examine effects on other channels. We really want to look at the mechanism of action of what's going on here, and also really try to mutate some of these residues that coordinate binding. And another thing that we started on as well is to really start targeting 6.1, looking for potential openers of KR 6.1, because I think therapeutically that could be more interesting than 6.2. And that's all I've got to say. Thank you.
Thank you very much. Any questions?
Thanks, William. Do you think 2985 is in competition with penicidil in the binding sites?
I do not think so, no, because penicidil binds to the SUR subunit and not to the KR6 subunit. Mm-hmm. This one seems to bind specifically to the KR6.2 subunit and not the SUR. So we haven't actually looked to see if there's competition between the two, but I don't think there's going to be a competition to the same binding pocket, if that's what you're asking.
Mm-hmm. But if you do some sort of dilution assays with the two compounds, you might be able to see.
You might indeed. Oh. We haven't done that.
Okay, thanks.
Oh, there comes trouble.
That was lovely. It's lovely.
Thank you.
I'm just a couple of questions. First of all, you are applying it to excise patches, and it's going to get to the site, the binding site for PIP2, which is on the cytoplasmic side, if it's going to do that. How lipophilic is this thing, and how does it get there when you apply it outside cells?
So I do not know. It has to be lipophilic because, as you've seen in that one whole cell experiment, immediately after the application, actually, I'm going to go back to that.
Is it charged? PIP2 works because it's very negatively charged, and the head group, IP3, would never cross the membrane.
Yeah, so I really can't talk about this, whether it's charged or not, because I just don't know, Colin. Tim is here, and we can corner him and ask him that question.
Okay, so maybe it's sterically doing what PIP2 does without negative binding.
It does appear to be membrane permeable, because here you can see that the compound was applied at fairly high concentrations, and within about 10 seconds, there was activation of the channel.
The other question I have is to do with how specific it necessarily is. Because thinking about the examples you showed, so 6.1, there is some evidence in the literature that it's actually insensitive to PIP2 anyway.
Yes.
So that would be a reason why it wasn't. KR2.1 has a very high open probability normally.
Yes.
So it won't be activated by PIP2. So you might want to test decreased open state stability mutants
in KR2.1.
Or deplete PIP2 on KR2 patches and
Right.
see if it'll come back.
Yes, no, that is actually a really great idea. And probably the same with KR3.1 as well, because it has a fairly natural high open probability. About the 6.1 is interesting, right? Because if you look at Shaoling's latest structure of 6.1, it appears that the C-terminus is displaced intracellularly by a little bit relative to 6.2. And so the distance between a PIP2 binding and the membrane is larger, which is probably why it doesn't really interact with the membrane and hence not activate the channel. When I do in silico docking, and Tim will have to help me out and so we can do it properly, I do find that this compound actually binds to 6.1, but it doesn't increase the open probability. And it's probably the similar mechanism that we have with PIP as well.
Hi, William. Quick question that might help with the penicidil question. Have you tested on the delta C 6.2, which can express on its own, right?
Yes, interesting question. When you delete the last 40 amino acids of KR6.2, you basically delete what's called the ER retention signal, and 6.2 supposedly can go to the membrane by itself in the absence of SUR. Connor's question is whether that channel, in the absence of SUR, can be activated by the compound. And the answer is no. It actually inhibits the channel, a small inhibition, at 100 micromolar. So SUR is necessary it seems. We don't quite know why yet. It's possible that it could be binding of the compound to SUR1 as well. We don't know that yet. Yes, it is. Yes, it is.
Well, great. Thank you very much.
Thank
It's a
you.
great talk.